How does antigen cross-reactivity happen? Imagine your immune system as a highly trained security team. Sometimes, this team recognizes different intruders because they look similar. That's what happens with antigen cross-reactivity. When the immune system encounters an antigen, it uses special receptors like antibodies or T-cell receptors to identify specific parts called epitopes. These epitopes are tiny regions on the antigen that the immune system targets. If two different antigens have epitopes that look alike in shape or chemical makeup, the immune receptors might mistake one for the other. This means that antibodies or T-cells designed for one antigen can also bind to a different one that shares similar features. This process begins when lymphocytes, which are immune cells like B cells and T cells, recognize epitopes. Each lymphocyte has receptors that are specific but also flexible enough to recognize related epitopes. This flexibility is necessary because the immune system needs to respond to many pathogens with a limited number of receptors. For example, a single T-cell receptor can potentially recognize millions of different peptide major histocompatibility complex molecules, which are small protein fragments presented on cell surfaces. The similarity between epitopes depends on their amino acid sequences and their three-dimensional shape. Small changes in the amino acids can make the immune receptor less likely to bind, but if enough similarity remains, cross-reactivity can still happen. The immune response often involves many antibodies targeting different parts of an antigen, so cross-reactivity can vary depending on which epitopes are recognized and how well they fit. Cross-reactivity can be helpful because it allows the immune system to respond quickly to related pathogens. For example, if someone has been exposed to one strain of influenza virus, their immune system might recognize similar parts on a different strain, providing some level of protection. But it can also cause problems. If the immune system reacts to a new pathogen based on a similar but not identical antigen, the response might be weak or even harmful. Sometimes, low-affinity cross-reactive antibodies can facilitate the entry of pathogens into cells through a process called antibody-dependent enhancement. In summary, antigen cross-reactivity happens because immune receptors recognize shared or similar epitopes across different antigens. This allows the immune system to respond broadly but can sometimes lead to less precise responses. It's a balancing act that helps protect us from many threats while managing the limitations of receptor diversity and specificity.